Hello, my dear students. Welcome you all to Sri Jayendra Rank Zone classes. I am your mentor and your friend for the subject biology, and I will be taking the zoology section. My dear students, in the last lectures, in the last videos, we learn about different theories of organic evolution, that is the biological evolution. We learn about Lamarckism, Darwinism. We learn about neo-Lamarckism, neo-Darwinism. We learn about the differences between the contradictory points between both the theories of Lamarckism and Darwinism. So in the consecutive lectures, we will be discussing about the mutational theory, which was given by Hugo de Vries. Okay. Before that, I will be telling some more concepts which will clear you, which will help you to fight your competitive exam. Okay. So first, the time is for Neet time, that is not the tea time. Okay, so we are basically going to solve some questions. The first question is genetic drift. You know about genetic drift? I explained. Genetic drift, it operates only smaller population, larger population, Mendelian population, and island population. So when we were learning about the genetic drift, we came to know that genetic drift involves the smaller part of the population because it is easy for the smaller part of the population to come up with the new change, to come up with the new character. And that is why the correct answer will be smaller population. The genetic drift operates only in the smaller part of the population, right? Second question is homologous organs. We learn about evidences of organic evolution, in which we learn about evidences from the morphology and the anatomy, in which the homologous organs are the organs which share same origin, same structure, and same function. Sorry, different function. Same origin, same structure, but different function, right? So homologous organs are the organs which share same origin, same structure. So the first option is wings of insect and wings of bat. Okay. Second option is gills of fish and lungs of rabbit. Pectoral fins of fish and forelimbs of horse. Wings of grasshopper and wings of crow. So now, the first option, wings of insects and wings of bat. Wings of insects are rudimentary, but they are basically for the flight. And bat also, the wings, they are for flight. Gills of fish and lungs of rabbit, they are having a common function. That is, they can be the part of convergent evolution or the analogous organs, but they are not homologous because their function is same. Respiration. Pectoral fins of fish and forelimbs of horse. Yes, this is the correct option. Pectoral fins of fish function to swim. Forelimbs of horse for running. Different function, but they are having same origin. Okay, so pectoral fins of fish and forelimb of horse, they share the same ancestor. Okay, they share the same ancestor. That is why they can be considered as homologous organs. Now, peripatus is a connecting link. You know, peripatus is a connecting link between annelida and orthopoda. So, peripatus, it is a connecting link between annelida and orthopoda. Now, next, convergent evolution. Convergent evolution is illustrated by dogfish and whale, rat and dog, bacterium and protozoan, starfish and cuttlefish. In the examples I have given, the convergent evolution, which shares different ancestry, but they have same function. And the right option will be that dogfish and whale, they perform same function. Okay, so their functions are same, and that is why they are at a part of convergent evolution. The diagram below, it is not below, it is above. The diagram below shows four species of birds that evolved from an ancestral species that had a small pointed beak. Okay, I mean ancestral species was having a small pointed beak, Today, all four species inhabit the same island. Okay, they are living in the same area. Which statement best explains the variation in the beaks of these four species? Okay, so previously the beak was small and pointed. Now the beak shape is changed. Why? You can see the size of the beak is increased. And why it happens might be there will be the or there was the plenty supply of food. So to actually capture more number of grains, okay, might be the size of the beak can be increased. So we'll see to the options. Over time, an abundance of seeds for food lead to increased similarities. The option is wrong. Increased similarities, no. Over time, an abundance of seeds for food lead to increased differences between the species. Okay, mean these can be a part of Darwin finches, right? So over time, an abundance of seeds for food lead to increased differences between the species. So these differences between the same species are because of the adaptive radiation, right? Adaptive radiation. Okay, Darwin finches, right? So option B is the correct option. Okay, when option B is the correct option, C and D, competition of limited food sources, they can lead to new speciation. Okay, competition of limited food resources lead to the selection of different traits, that also will give to a new speciation or new species. So option B is the correct option. Because of the large amount of seeds, they have to actually increase the beak size, right? Because ancestors, they were having small and pointed beak. Understood? Now we are moving to our topic, natural selection. If this is the part actually we were doing the last time, okay? We actually have done the bottleneck effect, the founder effect, and that part only contains the natural selection. If differential reproduction, some individuals produce more and some only few and still others none, continue for many generations, genes of the individuals which produce more offspring will become predominant, and that's very obvious in the gene pool of the population. Okay, when there can be some part of the population which is not reproducing, not reproductive health, they are not reproductive fit. Some of the members, they are very high on the reproductive fitness, they are very high on the reproductive fitness, they can give to large number of offsprings, and some of them they are giving average number of offspring, some of them they are giving less number of offsprings, and some of them they are giving no offspring. Okay, so the fact is the one which is giving more number of offspring will be predominant in the gene pool. Okay, means the number of that offspring will be high, right? And that is why it is known that the predominant species is the one which will dominate on the resources available at that particular area, dominate on the space of that particular area. So space, shelter, and food, these are some common, parameters on which the dominancy or the natural selection actually depends. Thus, the natural selection occurs through differential reproduction in the successive generations. That is very important. Okay, so natural selection actually occurs due to differential reproduction. Differential reproduction means the reproductive capacity of different members of the same group. It basically decides that what will be going to be next. Understood? Now, isolation. Isolation is a segregation or the separation of the populations by some barriers. These barriers can be physical barriers, they can be reproductive barriers which prevent interbreeding. So, isolation basically, it can be of physical type, mean physical barriers. Okay, when mountain is in between the troops of two in population, and nobody can cross through that mountain, and that is why there is a physical barrier or a climatic barrier. Okay, And so all these barriers, they actually prevent interbreeding, right? And because of this lack of interbreeding, the reproductive isolation, it occurs. The reproductive isolation between the populations due to certain barriers lead to the formation of new species. Okay, because of the species which is on the lack regarding the resources, it will adapt according to the natural selection, and it will may come up with some new characters, and those new characters, they can be transformed to the next coming generation, and that is why they can be a part of pioneer of new species. Okay, so the isolation is a segregation of the population by some barriers which prevent interbreeding. The reproductive isolation between the population due to certain barriers leads to the formation of new species. So these are some common, points which actually leading to the formation of new species or speciation. Now coming to next, industrial melanism. It's a very important phenomenon. This phenomenon was studied by Bernard Kettlewell. Figures showing white-winged moth and dark-winged moth in the next slide. Dark winged moth is basically melanized, mean more amount of production of melanin, pigmentation on the tree trunk in unpolluted area and in polluted area respectively. The white winged moth is basically in the unpolluted area, and the dark winged moth is in the polluted area. Right? I'll change the ink. Understood? So the in the unpolluted area, it is the presence of white wing, okay, having the required amount of melanin production, and in case of polluted area, it is dark winged, okay, because of the more amount of melanin production. Before industrial revolution in European countries, in Europe, cities of Europe, when the dull gray forms of peppered moth, Biston bitularia, sorry, were dominant. Okay, so peppered moth, the dull gray forms were dominant before the Industrial Revolution provoked in the regions of Europe. This Biston bitularia, the dull gray form, was actually dominant. The Carbonaria form, that was black, was rare because it was susceptible to predation by birds. As it was dark, so predators can easily capture the sight of the prey, and they basically come to attack, to predate those moth. Okay, so birds basically come to have these Carbonaria form, which were black, okay, which were totally contrasting on the green color, green color layered of lichens or lichens on the trunk of the tree. Suppose I'll make a trunk of the tree. This trunk of tree was basically covered by the greenish layer of lichens, and this green layer of lichens was actually covering the whole of the trunk. Now if a black colored moth is present on that area, okay, so that black colored of moth can easily be sighted by the predator birds. So predator birds can easily come and capture the prey, and that is why the Carbonaria form was actually rare. Okay, now the industrial revolution resulted in large scale of smoke, large scale of production of exhaust, which got deposited on the tree trunks. Lichens or lichens, they are biomarkers for pollution. Biomarkers for pollution, right? So because of large amount of production of smoke, the lichens, their color actually changes. Okay. So because of industrial evolution resulted in large scale of smoke which got deposited on tree trunks, turning them into black. Previously they were green or yellowish green. Now they are black. Now gray varieties become susceptible. Now the gray colored moth, distant butiraria, which was the commonest form, now it becomes susceptible. Okay, but the Carbonaria form it got the survival advantage. So here I'll write the Carbonaria form, it got the survival advantage. Okay, it got the survival advantage, right? So now what happens? The black worms flourished. So in this case, the black worms which were previously very rare, now they actually are going to flourish because their competitors, their struggle was actually reduced. And this is what the natural selection. The natural selection gives a source of survival advantage to the Carbonaria form. Replacement of coal by oil. Okay. Then in the industrial evolution, coal was replaced by oil and the electricity reduced the production of black moth. So the frequency of the gray moths now increased. Okay. So just after reaching to the peak of industrial revolution, the coal was actually replaced by oil and the electricity. Okay. In the mid of the 18th to the 19th centuries. So what actually happens? The black moth again starts to reduce and the gray moth again starts to increase in their frequency, in their, the population size. Okay. So this basically gives a help to connect. Okay. Industrial melanism is basically based on the parameters of natural selection. The one who got the survival advantage will be considered as a survival or a fit for survivor, means survival of fittest. Now coming to next, drug resistance. The drugs which eliminate pathogens become ineffective in the course of time because those individuals of pathogenic species or the pathogenic, varieties, they develop resistant factors. Okay. Now they survive, flourish to produce their tolerant population. Like for example, DDT. DDT was actually used as an anti-malarial agent. But sooner or later, the protozoan, which was responsible for the malaria, it actually come up with the resistance power against DDT. Okay, DDT was also used as a fertilizer. So DDT, the resistance developed against DDT basically gives a natural selection or gives a survival advantage to the protozoans. Okay, tuberculosis. Now tuberculosis, TB, also shows multiple drug resistance. MDR, this multiple drug resistance. This multiple drug resistance basically it tells us what? That okay, if we are giving only one drug again and again for a very long time, then what is going to happen? That drug basically becomes inactive or ineffective because of the bacterial plasmid which undergoes some kind of mutations which will lead to come up with the antibiotic resistance, okay, or the drug resistance. So this drug resistance is basically the part of the historical, the part of the medical history, that how can be a protozoan or a pathogen, okay, it can behave smartly to come up with the effect of that particular drug or the antibiotic. So all these industrial melanism, reproductive isolation, drug resistance, they actually the subparts or the examples which prove the natural selection. Okay, this is the industrial melanism. Okay, now you can see the deposition of the this black color. Okay, of the or we can say the smoke. So you can see the peppered color, the dull grayish color. Now it becomes what? It becomes what? Contrasting. So previously it was not contrasting. It matches the color of the tree of the trunk, and this was the actual contrasting color. Now This is the contrasting color. So this is what the survival advantage. Okay. Examples of natural selection. Sickle cell anemia. Individuals which are homozygous for sickle cell anemia die at an early age. You can say, or you can see the sickling gene which causes the distortion of RBC, red blood corpuscle, because of gene mutation. So the persons which are homozygous mean if they are carrying a pair of sickling gene, okay, so they die at an early stage because in case of homozygous condition, it becomes lethal. It becomes lethal gene or lethal effect. In case of homozygous condition. In heterozygous individuals, they become a carrier. The cells containing abnormal hemoglobin, sickle shaped. In case of females, it is carrier. In case of males, it is symptomatic. Okay. So in case of males, I'll write here. It is symptomatic. And females are carriers, heterozygous. Now males become symptomatic means what? If they are having the HBS or the sickle cell trait, means the females having sickle cell trait, but the males they are having only one X chromosome, and that is why if that X chromosome is having the sickle cell gene, then it becomes symptomatic. The sickling of RBC reduce the oxygen tension, and as the oxygen carrying capacity will reduce, what will happen at the high altitude regions they are going to suffer, and that is why the males become symptomatic. So in heterozygous individuals, the cells containing abnormal hemoglobin means becomes sickle shaped. The biconcave shape becomes sickle shaped. Okay. In fact, when the RBC becomes sickle shaped, it kills malarial parasite. Now, how the native people of Africa, okay, when just around the equator regions, they become or they got a survival advantage of because of sickle cell trait or sickle cell anemia in case of males. As the sickling of RBC reduces the hemoglobin content. So the moment hemoglobin content reduces, the malarial parasite cannot come up with its complete life cycle. So the malarial parasite cannot, complete its erythrocytic zygogony, which basically affects the life cycle of malarial parasite. And as the life cycle of malarial parasite will be reduced, will be disturbed, it cannot withdraw the nutrients because the hemoglobin content is less, and that is why the malarial parasite dies effectively. So that these individuals area able to cope up with malarial infection much better than the normal person. So being a sickle cell RBC, being carrying the sickle cell RBC, they got a survival advantage against malaria. The process of natural selection thus maintains the abnormal form of hemoglobin along with the normal form in a region where malaria is. So wherever the malaria is common, the sickling of RBC can be seen along with the normal biconcave shaped RBC. It is a survival advantage in case of sickle cell anemia in case of males and sickle cell trait in case of females. Now malaria and G6PD deficiency, glucose 6-phosphate dehydrogenase enzyme, glucose 6-phosphate dehydrogenase. Dehydrogenase enzymes always initiate the activities of nicotinamide adenine dinucleotide phosphate reduced form. Okay, so reduced form means it is having a reduced form. Mean it is a either it is accepting the electron or it is accepting the H plus sign. So I'll write NADPH. So whenever the dehydrogenase enzyme is basically involving in any metabolic reaction, you just have to have, I mean, you just, that this glucose 6-phosphate, it basically activates the NADPH, reduced nicotinamide adenine dinucleotide phosphate. Now, During the process of glycolysis, this dehydrogenase enzyme helps to convert glucose 6-phosphate into other compound. Now, glucose 6-phosphate dehydrogenase enzyme deficiency, the enzyme becomes deficient, is a common abnormality in negroids, in Negroes. Hemoglobin gets denatured. Okay, why hemoglobin? Hemoglobin, it is a a metalloprotein. It is a metalloprotein, and the denaturation of protein basically tells us that okay, conditions were not appropriate, not optimum. And why? The fact is, RBCs, they don't have mitochondria, they only have cytoplasmic content, and that is why the only source for them for the production of ATP is glycolysis. And if the glucose 6-phosphate dehydrogenase enzyme, it is decreased or it is not secreting in the required level, then what will happen? The glucose will not be converted into the other forms. Okay, so this glucose 6-phosphate dehydrogenase deficiency basically stops the metabolism of glucose into the pyruvic acid. Okay, and the ATPs will not be produced, the energy carriers will not be produced, and that is why the hemoglobin gets denatured and is deposited on the cell membrane. So the deposition of hemoglobin in the cell membrane basically leads to the basically it is acting as a survival advantage of the person suffering from malaria. Okay, this disease is also known as favism. In these RBC, the malarial parasite cannot complete its life cycle. Okay, such person get protection against malaria. So G6PD deficiency basically becomes a survival advantage, as in case of sickle cell anemia also becomes a survival advantage of the person suffering from malaria. As malarial parasite, for the completion of its life cycle, it needs what? Glucose to be converted into the next metabolic product. But the fact is, because of G6PD deficiency, it is not happening. And that is why the hemoglobin gets denatured. It gets stick on the cell membrane. And that is why the person suffering from malaria, they actually come up with, good amount of survival rates. They come up with the survival advantage. Okay, this is known as what? Favism. Okay, so sickle cell anemia and malaria and G6PD deficiency, these are the common examples. Now coming to next, artificial selection. As we have studied natural selection, now we are going to move on with artificial selection. Man has been taking the advantage of genetic variations for improving the qualities of domesticated plants and animals. Okay, production of high yielding varieties. Okay, so production of high yielding varieties basically helps to HYV. Okay, the production of high yielding varieties, they actually involves the inoculation or the administration or artificially giving them a survival advantage by getting them genetically changed. Okay, so that is why the men come up with these genetic variations to control the good qualities of domesticated plants and animals. He selects the individuals with desired traits, desired characters, and separates them from those which don't have such characters, means segregation. This segregation can lead to reproductive isolation. The selected individuals are interbreeded. Now, the selected individuals, they are interbreed to get a good quality. This process is called artificial selection. This process is man-made. Okay. If it is represented by many generations, it produces a new breed. Obviously, it is going to produce a new breed with desired characters. By artificial selection, animal breeders, they are able to produce improved varieties of domesticated plants and animals like dogs, horse, pigeons, poultry, cow, goats, sheep, and other. Okay, from the wild ancestors. Wild ancestors means the normal variant. Okay. Similarly, the plant breeders, they have obtained improved varieties of useful plants like wheat, right? During Green Revolution, which was introduced by M.S. Swaminathan in 1992, but actually it was a phenomenon which evolved, around 7 months back or years back by Norman E. Block in the Europe. Okay, So they actually have produced semi-dwarf varieties, the varieties which actually require less amount of minerals, less amount of raw materials. So the soil fertility can be maintained for a longer time. With these semi-dwarf variety, they actually come up with very large amount of production of grains. So wheat, rice, sugarcane, cotton, pulses, vegetable, fruits, they all are basically having or introduced by all these genetical variants to improve their qualities. Artificial selection is similar to natural selection, except that the role of nature is taken by man. Okay, and the characters selected here are of human use. Okay, the breeders have successfully produced toy-like Shetland pony, the Dane dog, the sleek Arabian race horse by the selection. Because of artificial selection, they actually have produced all these varieties like Arabian's racehorse, Dane dog, etc. Many crop plants like broccoli, kale cabbage, cauliflower, Brussels sprouts, kohlrabi have been produced, okay, through selective breeding. So that is known as what? Artificial selection. According to the need, the varieties are improvised. According to the need, the varieties are improved. So that is why it is known as what? Artificial selection. Understood? The various breeds of fowl, ranging from the ceremonial cocks, the Japanese Wangodorai, to the broilers. Okay, mean the broilers, they have been basically introduced by these genetic variants to come up with large amount of flesh production or meat production. Understood? And that is why they are known as what? The improved varieties. Okay, leghorns are also derived from a single jungle fowl, Gallus gallus. So leghorns, exotic varieties or improved varieties, they come up with very drastic and good characteristics as in compared to the wild varieties, and that is why it is known to, or it is good to use them for the commercialization. Understood? Now coming to next, we are having, or we are moving to, yeah, these are breeds of domestic pigeon from which are basically formed from the wild rock pigeon. Okay, these different rock pigeon. This is the rock pigeon. I mean, this is the wild variety. Just a minute. This is the wild variety, and it is produced. It is basically artificially selected to give rise to different varieties. Okay, Fentac, Porter, Jacobin. So all these varieties are artificially selected, right? Artificial selection basically helps to continue the desired character without fail. Okay, although failures do happen, but the chances of continuing the desired characters are high in comparison to the natural selection. Now is reproductive isolation. Okay, reproductive isolation is the prevention of interbreeding between the populations of two different or closely related species. So it is basically a prevention of interbreeding between the population of two different or closely related species. They are not able to reproduce. It maintains the characters of the species but can lead to the origin of new species. It maintains the characters of the wild varieties, the inhabited varieties, but it may lead to the origin of new species. The mechanism of characters of the species but can lead to the origin of new species. Mean the mechanism of the formation of new species, it may lead, or it may is having basically based on reproductive isolation as well. Okay, The mechanism of reproductive isolation is explained by Stebbins in his book Process of Organic Evolution. So Sir Stebbins, he proposed process of organic evolution to basically tell us that okay, reproductive isolation, it can be a method to have a new species. Ecological isolation. Okay, there are mainly two subtypes of reproductive isolation. First is the pre-mating or the pre-zygotic isolation. So basically, I think I have forgotten to write it. Pre-mating or pre-zygotic. So this is the pre-mating or the pre-zygotic isolation, which includes ecological isolation. Two species live in different habitats, areas, and do not meet. They don't meet. One may be living in fresh water and the other in sea. So as the habitats are different, they are not going to meet. And that is why they are basically leading to reproductive isolation. And this is the ecological isolation because of the climatic barrier, ecological barrier. Okay. Temporal isolation. Breeding seasons of flowering time may be different in the two species. Breeding seasons or the flowering time may be different in the two species. Okay, in botany you have learned about the protoentry and protogyny. Okay, these different terms. Okay, that the anther, they are basically common. They are getting matured first, or the gynoecium is getting matured first. But the fact is, at the same time, they need to mature, okay, for the fertilization and all. That is why breeding seasons, if they are different, okay, of two species, mean they are not going to disperse their seeds to one another. The cross pollination or the cross fertilization chances are very reduced. So temporal isolation basically it decreases the frequency of cross breeding. Cross breeding. Behavioral isolation. The males of one animal species are unable to recognize the females of another species as potential mates. Okay. With males of one animal species, they are basically unable to recognize the females of other species as their right co-partners. Okay. This is behavioral isolation or psychological isolation. Mechanical isolation. The structural difference in the genitalia, genetical organs, of individuals belonging to different animal species interfere with mating. Okay, these are some of the barriers. Okay, like mechanical barriers. The genitalia are different, and that is why the sexual intercourse could not happen. Okay, because of this mechanical isolation. Then the gametic isolation. The sperms and ova of different species of animals are unable to fuse. Gametic isolation. The sperms and ova of different species are unable to fuse. Okay, these are gametic isolation. In plants, the pollen coming from a different species may be rejected by the stigma. Stigma will not let that pollen to come up with the pollen tube. It will not germinate the pollen because the process is, it is a chemical process, a chemical movement. Okay, so this chemical movement is involving the secretion of some sugary like substances on the surface of stigma to attack the pollen for their germination. And if it is not happening, it means pollens are not compatible. So this is known as what? Pollen incompatibility. Pollen or sexual incompatibility. Understood? So these are different types of reproductive isolation, and what exactly reproductive isolation is. We have covered the first type of reproductive isolation, which is pre-mating or pre-zygotic isolation. Now the second, the post-zygotic isolation, in which a hybrid zygote is formed, but it may not develop, may not flourish into a viable or fertile adult. Hybrid zygote is formed, okay, when pre-zygotically they were not able to isolate them. So they come up with the, formation of zygote, but that zygote cannot develop. At that zygote, may it lack some of the essential genes or the information because of this crossing over and all, and that is why they cannot develop into viable fertile adult. Hybrid inviability. Hybrid zygotes fail to develop in plants. Embryos arising from interspecific crosses abort. Okay, if we are going for interspecies cross, okay, two different species members, they have been crossed. Okay, so after they cross, they become inviable, mean or inviable, mean they are not of any use. Okay, these crosses, they get aborted, the embryonic development get aborted. Hybrid sterility. Hybrid adults do not produce functional gametes, mean they are sterile. Mules, henny are common example that they are sterile. They are produced, okay? They are produced for the fulfilling of a particular desire, but the fact is they are not fertile in mammals. Several hybrid ornamental plants are sterile. There are many ornamental plants which are sterile, okay? Hybrid breakdown. Some interspecific mating produce a hybrid, okay, which give rise to next hybrid by backcross, but they have reduced vigor or fertility or both. Okay, so the fact is, hybrid vigor is simply mean if the hybrid variety is having the capacity of being fertile, okay, or production of gametes, that is hybrid vigor. But the fact is, these hybrid vigor is very less to actually function on a normal scale, as the interspecific genes present in them, information present in them, maybe can lead to the production of some abnormal function, and that abnormal function could interfere with their fertility part. Exceptions: African lioness and Asian tiger, Panthera leo, Panthera tigris, they form tigon. Okay, male lion, female tiger, liger. Polar bear, Alaskan brown bear, okay, they are also fused. Mallard duck, pintail duck fused. Platyfish, swordtail fish. So ultimately, you can see these are so many different we can say that transgenic animals which are prepared, offspring which obtained from all these hybridizations are fertile, but these species do not interbreed naturally. They don't need, they don't feel like to interbreed naturally, and that is why they have been enforced by the breeders to help them breed. Okay. Now coming to next, genetic basis of adaptations. What are the different genetic bases of adaptations? Joseph Joshua Lederberg and Esther Lederberg shown genetic basis of adaptation. That adaptations and adaptability, range of adaptability is having the genetical connection by experimenting on bacteria. Okay, this experiment is shown as Lederberg's replica plate experiment. Lederberg's replica plate experiment. According to Lederberg, Joshua Lederberg and Ethel Lederberg, they cultured the bacterial cell on the medium, agar medium. I'll change the ink. Cultured the bacterial cells on the agar plate, okay, which is the culture medium. Many bacterial colonies or groups grew on these agar plate, means culture medium. In this, every colony is formed by the division of bacterial cells. Therefore, it is all cells were of the same genetic structure. This type of group of cells known as clone. Okay, when they basically lead to the growth of the bacterial cells in vitro by putting them into a agar plate. This agar plate is having the essential nutritive supplementation for the growth and the division of bacteria. Now they come up with the fact that whatever the generations which are formed, they are of same genetic structure. Okay, and if they are having same genetic structure, the fact is they are having same ancestry. This multi-colony agar plate is known as master plate. Okay, so this is known as what? Master plate, which is having multi-colony of agar. Okay, having multiple colonies of bacteria. On this master plate, one sterile velvet plate was pressed. Okay, when on this master plate only having multiple colonies of bacteria, one sterile mean non-contaminated, not having anything. Okay, velvet plate was pressed slightly so that some bacteria got stuck on the velvet plate. In this way, this becomes a replica or the master plate, replica of master plate. Now, efforts of preparing a replica had been made on those agar plates whose agar contains an antibiotic penicillin. Now, the fact is this velvety plate is basically pressed on the master plate. The bacterial culture colonies, they got stuck on the velvet plate, and now they are going to be grown on the penicillin aided medium. Okay, it was seen that some bacteria failed to grow on the penicillin agar plate while some bacteria were able to grow and develop a new colony. Okay, it was concluded that these bacterial colonies were penicillin resistant. So that is what actually the drug resistant I was telling you. This bacteria have penicillin resistant mutant gene because of the presence of the plasmid. Understood? So they come up with the fact that they are penicillin resistant. Why they are penicillin resistant? Because of the presence of extra chromosomal material plasmid, they have that penicillin resistant gene in them. Now, what was the Lamarckian view on this? Penicillin induced a change in some bacterial cells, enabling them to grow in a medium containing penicillin. That was a wrong concept. Okay, actually, the bacteria, they come up with some mutations, genetic mutations, changes in their DNA, which could lead them to produce a agent or a substance which could break the structure of penicillin, or which could survive in the presence of penicillin. According to Darwin, some bacteria were penicillin resistant in the bacterial suspension. In penicillin medium, normal bacteria did not survive, while the mutant bacteria survive as they are adapted and form colonies. Okay, so that was the genetic basis of adaptation. That upcoming of the new species speciation is basically having some genetic, background. Okay, so that seed was actually given by Lederberg's replica plate experiment. Okay, I hope is this clear. Now moving to the next, modern synthetic theory of organic evolution. This theory is the result of the framework of a number of scientists, namely Dubinsky, Fisher, Haldane, Solright, Mayer, and Stebbins. Stebbins discussed his theory in his book Process of Organic Evolution, and Dubinsky explained in his book Genetics and the Origin of Species. Okay, so Stebbins in his book Process of Organic Evolution, and Dubinsky explained in his book the Genetics and the Origin of Species. Understood? In the reproductive isolation, when I was addressing the previous slides of reproductive isolation, the reproductive isolation was actually, the mechanism was explained by Stebbins only in his book Process of Organic Evolution. Now coming to next point, according to this theory, new species cannot evolve by the presence of variable genotype in a population. If a population is having different types of genotype, variable genotypes, new species cannot evolve. Two factors are always required, that is natural selection and reproductive isolation. There should be a need for the formation of new species. My dear students, please remember my point. There should always be a need for the formation of new species. If there is no need for the formation of new species, it won't evolve. Okay. So reproductive isolation and natural selection, these are two factors which are responsible for the upcoming of new genotype. Natural selection guides different population into a different adaptive direction, and reproductive isolation between them due to geographical barriers leads to these direction to evolution of new species. How these work? That natural selection guides different population into a different adaptation, that the members of the different population, they come up with some certain changes which could give them the survival advantage, and the reproductive isolation, they come up with the geographical barriers in which they actually have only one way to reproduce and to form new species. In this theory, following factors are included: gene mutation. Okay, so maybe the reproductive isolation can lead to the changes in the DNA of the existing or the survivor species, and the natural selection too, and changes in the number of chromosome and the structure. Changes in the number of chromosome and structure. Genetic recombination, hybrid, natural selection, and reproductive isolation. So in this theory, modern synthetic theory, all the concepts were there: genetic mutation, change in number of chromosome and structure; genetic recombination means the formation of hybrid DNA; natural selection; and reproductive isolation. Besides these factors, there are two main processes which can cause evolutionary change. Apart from these two main factors, there are two main processes which can cause evolutionary change, and these are migration of individuals from one population to another. The migration of individuals basically introduce new genes, introduce new genes in the gene pool, right? So the frequencies of having new genes can lead to the formation of some new characters and may lead to the organic evolution. Hybridization among some species mean mating, making of the hybrid vigor, okay, or the genetic recombination. Also related genera, which causes genetic variation in a population undergoing process of evolution. Closely related genera, closely related members, they can actually undergo the process of overruling the barriers of reproductive isolation. They can interbreed, and because of this interbreeding can lead to genetic recombination and the formation of new species. Okay, so I hope this modern synthetic theory, which was actually proposed by Dubansky, Fisher, Haldane, Swellbright, Mayer, and Stebbins, mainly Stebbins and Dubansky. Okay, Stebbins proposed his book Process of Organic Evolution, in which he mentioned actually the modern synthetic theory, and same thing was done by Dubansky. According to this theory, there are two, if the new species, it cannot evolve if there is no need, there is no desire. Okay, so two factors are always responsible for the formation of new species, that is natural selection and reproductive isolation. Along with these factors, certain more factors like genetic recombination, gene mutation, change in number of chromosomes and their number. Okay, along with migration of the individuals, which come up with some new genetic variants, genetic frequencies in the gene pool, and the hybridization. Okay, so this topic, I think modern synthetic theory is covered and cleared. Now moving to next, mutational theory. It was proposed by Hugo de on the basis of his experiments on the plant Oenthera lamarcana, means evening primrose. Okay, Oenthera lamarcana, which basically commonly known as the evening primrose, was the experimental tool of Hugo de Main point of mutational theory put by Hugo de were: mutation is a discontinuous variation, and it is a raw material for evolution. That also it's very clear that mutation is a discontinuous variation and it is a raw material of evolution, because mutation will happen or not, you actually don't know. Mutation appears suddenly, okay, and produced their effect immediately, means random. Okay, mutations are different from the parents and there are no intermediate stages between the two. There is no link or linkage, no linkage. When the parental characters, they are entirely different from these recombined factors, recombined characters, because of mutations. The same type of mutation can appear in several individuals of a species. In a particular species, the same type of mutation can happen in several individuals. Mutation can appear in all directions, okay, and all mutations are inheritable as they are involving change in gene frequencies. Okay, and that is why they are inheritable. Understood? Now, useful mutations are selected by the nature under the process of natural selection, and lethal mutations are eliminated. The mutations which are helpful for the survival of the new species or for the formation of new species, they always survive. They always dominate, but the lethal ones, they actually abort themselves. Okay. Mutations are recurring so that they have, same mutant effect, okay, can appear again and again. So change of selection by nature are increased and new species is formed. Mutations are always recurring, it always happens again and again, so that it can lead to the formation of new species. Okay, DeVries termed single step large mutation as saltation. Single step large mutation means which is basically involving the more population size, it is known as what? Saltation. So according to De Vries, if a single step large mutation is happening, it can lead to saltation. Mutations are large, random, and directionless, while Darwinism variations are small and directional. The difference between mutations and the Darwinism variations were that the Darwinism variations, they were small and directional, but the mutations, they are random and directionless. So this is the mutation theory which was proposed by Hugo de Vries on the basis of his experiment Oenthera lamarckina, evening primrose. Main point put by mutation theory were that mutation was discontinuous, it can happen anytime, it is random, and it shows sudden, sudden effect, immediate effect. Mutants are different from the parents, and there is no intermediate stage to connect them. The same type of mutation can basically occupy or acquire many individuals of the same population. Mutation can appear in all directions as all mutations are inheritable. Okay. Useful mutations are selected by nature and lethal mutations are eliminated. Mutations are recurring, it happens again and again until the formation of new species. De Vries term single step large mutations are known as what? Saltation. Mutations are differ from Darwinism variations, that mutations are large, random and directionless, while the Darwinism variations are small and directional. Understood? That was the mutational theory. Now, what are the points in the favor of mutational theory? Mutations are actually the source of all variations and fountainhead of evolution. That's very important. Mutations are actually the source of all variations and fountainhead of evolution. Okay. Mutational theory can explain both progressive and retrogressive. Progressive means from simple to complex, and retrogressive means from complex to simple. Understood? So progressive evolution means in which the simple form is basically evolving into the complex form, and the retrogressive means in the backward direction. The retrogressive form with the more complex form is going to form the simple form. This theory can explain both change and unchanged forms, whether the form is changed, okay, or the form is unchanged. This theory, mutation theory, was the only theory which was able to explain that whether this is change, mean a mutant or the normal form. Significance. Hugo de Vries' mutational theory generally accepted because the mutations were found to be inheritable. That's very important point. It was later through that evolution cannot occur by mutation alone. Natural selection and reproductive isolation of mutants are also necessary for evolution. So all the three factors, the cumulative effect is seen. Okay. Criticism. Oenthera lamarckina was actually not a normal plant. It is a heterozygous form which having chromosomal aberrations. Okay. Increase or decrease in total number of chromosomes, okay, from the normal variant. Natural mutations are not common, they are very rare. Most of the mutations are retrogressive, mean from complex to simple. Most of them are recessive, unable to explain mimicry and mutual dependence. So because of mutational theory, we got to know that actually mutational theory was unable to explain the mimicry. Okay, mimicry I will be telling you when we will be dealing with the biodiversity part. So mimicry, it is a process of adaptation. Okay, like in case of camouflage, you see. Okay, there are many species which basically mimic the natural environment. So mimicry was not explained by mutational theory. Mutual dependence was not explained by mutation theory. Okay, most of the mutations are retrogressive. Okay, and most of them are recessive. This is the fact that is why which imposed or proposed a criticism. Okay. Now we will be doing the natural selection and the polymorphism in the next class. Till then, my dear students, stay safe, stay at home. Bye bye. Take care.